culinary appeal of rhubarb is undeniable, yet the leaves function as a repository for oxalic acid. While a lethal dose theoretically requires consuming nearly 5 kilograms of foliage, the crystallization of this compound can precipitate kidney failure at significantly lower concentrations. Similarly, the husk of an underripe tomatillo masks defensive toxins that require precise timing to degrade. The critical task is distinguishing the edible harvest from the plant's chemical armory. Mastering this biological anatomy ensures that only safe components reach the plate, starting with a crop designed to weather the harshest winters. The potato functions as a biological battery, a dormant energy reservoir buried in the soil to survive the winter and fuel new growth in the spring. However, this global dietary staple possesses a latent chemical defense system that activates under specific environmental triggers. When the tuber leaves the darkness of the soil and encounters light, whether from the sun in the field or the artificial fluorescence of a storage facility, it interprets this illumination as a signal to prepare for defense against herbivores. This exposure initiates a rapid biological synthesis of glycoalkaloids, primarily a compound known as solanine. The plant effectively transforms from a passive carbohydrate source into a chemically active organism, protecting its reproductive future. This defensive shift manifests through a clear visual indicator. Simultaneous with solanine production, the potato acts to manufacture chlorophyll, the pigment responsible for photosynthesis. This causes the skin of the tuber to turn a distinct shade of green. It is crucial to understand that the green pigment itself poses no physiological threat to humans. Rather, it serves as a reliable proxy for the invisible accumulation of toxins. The intensity of the greening often correlates with the concentration of the defense chemicals. The potato uses this chlorophyll as a metabolic engine, but for the consumer, it functions as a visual warning flag that the chemical composition of the vegetable has fundamentally altered. A dangerous misconception persists regarding the preparation of these compromised tubers. While heat effectively neutralizes many bacterial pathogens found in food, solanine possesses a robust molecular stability that withstands high temperatures. The compound survives the thermal rigors of boiling, baking, and even deep frying. A green potato processed through a fryer remains a toxic potato, as the neurotoxin persists within the starch matrix. Ingestion of solanine poses a severe health risk, as the neurotoxin targets the nervous system. The biological objective of the compound is to punish predators, and in humans, sufficient quantities lead to systemic toxicity that mimics severe illness. The distribution of this toxin follows a specific anatomical pattern. The plant concentrates solanine primarily in the skin and the layer of flesh immediately beneath it, creating a defensive perimeter around the valuable starch inside. Consequently, the mechanical removal of the skin through peeling significantly reduces the total alkaloid load. However, peeling is not a guaranteed solution. Research indicates that the toxin permeates into the flesh, particularly in tubers that have undergone extensive greening or sprouting. In these advanced stages of defense, the solanine migrates inward, rendering the simple act of peeling insufficient to ensure safety. The sprouts themselves act as high-density clusters of glycoalkaloids, containing concentrations far exceeding those found in the tubers. Evolution has provided a secondary sensory line of defense for mammals. Bitterness. Solanine imparts a sharp, astringent flavor profile that serves as a final fail-safe against intoxication. This bitterness functions as an evolutionary deterrent, a chemical signal warning the consumer that the solanine levels have crossed the threshold from negligible to hazardous. If a prepared potato delivers this specific bitter taste, it indicates that the tuber creates a toxic environment that the human digestive system is not equipped to handle. The only safe protocol involves the meticulous excision of all green tissues and sprouts, or the complete disposal of the tuber if the greening is widespread. It is also vital to distinguish the specific behavior of the potato from its botanical relatives. While the potato belongs to the classification of nightshades, its reaction to light is distinct regarding food safety. Other members of the family, such as bell peppers and tomatoes, are commonly consumed in their green, unripe states without the risk of solanine poisoning. The potato stands alone in this specific volatility, requiring a strict adherence to dark, cool storage environments to suppress its natural defensive pathways. Safe consumption relies entirely on blocking this synthesis through proper storage and ruthlessly removing any tissue that shows signs of activation. By keeping the tuber in the dark, you prevent the dormant chemical factory from ever switching on. However, chemical defenses are not limited to the tubers buried in the soil. They also appear in the hard, dry seeds of the protein-rich legumes sitting in your pantry. Among these stored provisions, the red kidney bean contains one of the most potent plant toxins known to man, hiding a severe biological threat within a common pantry staple. While often viewed as a harmless source of protein, The raw bean possesses a chemical defense system capable of inducing severe symptoms with the ingestion of even a small quantity of raw beans. The culprit is phytohemagglutinin, commonly abbreviated as PHA, a specialized lectin that functions as a sophisticated biological weapon. Upon ingestion, this compound survives the initial stages of digestion and actively binds to the intestinal wall, recognizing specific carbohydrate structures on the surface of the gut lining. This binding action physically interferes with the body's ability to absorb nutrients and triggers a massive inflammatory response. The physiological reaction to this binding event is rapid and violent, often mimicking the most severe cases of food poisoning within just a few hours of consumption. The symptoms include nausea, uncontrollable vomiting, and diarrhea as the body attempts to purge the detected toxin. The potency of PHA is so significant that in controlled studies, raw kidney beans have been known to kill laboratory rats, a grim metric that underscores the extreme danger of consuming this legume without rigorous thermal processing. The line between a nutritious ingredient and a medical emergency is entirely defined by the application of heat. A catastrophic error frequently occurs in domestic kitchens involving the use of slow cookers. These devices, designed for convenience and long-term simmering, often fail to reach the critical temperature required to denature the PHA protein. Phytohemagglutinin exhibits a terrifying stability at lower temperatures. Research indicates that heating the beans to only 80 degrees Celsius, a common range for slow cookers, does not destroy the toxin. Instead, it generates a paradoxical effect where the toxicity actually multiplies compared to the raw state. This phenomenon, known as the slow cooker effect, turns a batch of improperly prepared beans into a concentrated biological hazard, far more dangerous than if the beans had been eaten raw. To effectively destroy the lectin and render the bean safe, a specific and non-negotiable thermodynamic protocol is required. The molecular structure of PHA is only irreversibly shattered when subjected to a rolling boil at 100 degrees Celsius for a minimum of 10 minutes. This rigorous boiling process provides the energy necessary to unfold the protein, destroying its ability to bind to the human intestinal tract. Any temperature below this threshold, or any duration shorter than this 10-minute window, leaves the defense mechanism intact and potentially amplified. This creates a sharp distinction between industrial and domestic preparation. Canned kidney beans found in grocery stores are universally safe because they undergo pre-treatment under immense pressure during the canning process. The industrial retorts used in canning factories achieve temperatures and durations that far exceed the minimum safety requirements, effectively neutralizing the PHA before the can is ever sealed. Dry beans, however, require the cook to replicate this safety standard manually. The process demands a strict hydration protocol followed by the application of high heat. The outcome is strictly binary. Properly prepared kidney beans are a superfood, rich in fiber and protein, with the toxin completely deactivated. Undercooked beans remain a potent delivery system for PHA, capable of sending a dinner guest to the hospital. The mastery of this ingredient requires acknowledging that the slow cooker is not a universal tool, and that certain proteins demand the violence of a boiling pot to become edible. While heat effectively neutralizes the chemical defenses of the bean, other garden staples require a different strategy, relying not on temperature, but on a strict anatomical separation between the nutritious stalk and the lethal foliage. Rhubarb exemplifies this strict division, presenting a botanical dichotomy that challenges the casual gardener. While the plant appears as a unified organism in the garden bed, chemically, it is segregated into two distinct zones of safety and hazard. The vibrant red or green petiole, the stalk prized for its tart contribution to culinary history, is a delight, while the broad, fan-like foliage serves as a concentrated repository for oxalic acid. This is not merely a bitter compound intended to discourage grazing, it is a potent chemical defense mechanism. Within the cellular structure of the leaf, oxalic acid forms microscopic, needle-like crystals known as raphides. While these structures can cause immediate physical irritation to the mouth and throat, the primary physiological threat arises after ingestion. Once absorbed into the bloodstream, the acid aggressively binds with systemic calcium to form insoluble precipitates. These accumulations can physically obstruct the renal tubules, leading to acute kidney failure. This process turns a nutritious ingredient into a medical crisis, utilizing a defense mechanism the plant has refined over millennia. To contextualize the risk, Toxicology reports indicate that the lethal dose of oxalic acid for an average adult is approximately 25 grams. Achieving this terminal threshold would require the consumption of roughly 11 pounds of raw rhubarb leaves, a volume that is physically difficult to ingest given the texture and taste. However, one does not need to reach the lethal limit to experience severe toxicity. Significantly lower quantities can induce nausea, respiratory distress, and permanent renal damage, making the complete avoidance of the foliage a non-negotiable safety standard. A persistent agricultural myth suggests that these toxins migrate from the leaf down into the stalk as the harvest season extends into late summer. Agricultural science has largely debunked this concept of active migration during standard growing conditions. The plant maintains a rigid chemical border between the solar panels of the leaves and the structural support of the stalk. There is, however, a critical exception ruled by thermodynamics. When the plant is subjected to a hard frost, the cellular integrity of the leaf ruptures. In this specific scenario, moisture and dissolved oxalates can retreat from the dying foliage into the surviving petiole. Consequently, stalks harvested after severe cold damage may contain elevated toxin levels and are considered unsafe. The preparation process, therefore, demands a ruthless removal of all green leafy matter before the crop ever enters the kitchen. This is not a matter of trimming for aesthetics. It is a sanitary requirement. The separation must be absolute, ensuring that the toxic leaves are immediately composted while the tart stalks are processed for cooking. Recognizing the visual cues of proper separation ensures that the harvest remains a source of nutrition rather than injury. Yet, this theme of highly toxic components hiding in plain sight is not limited to temperate garden perennials. It extends into the staple crops of the tropical world, specifically to a root that requires a complex, multi-stage chemical exorcism to become edible. Cassava serves as the primary carbohydrate source for over 500 million people, anchoring the diets of entire nations across the tropical belt. This agricultural dominance is derived from the plant's extraordinary resilience. It withstands severe drought and aggressive pest populations that would obliterate less fortified crops. However, this survival capacity is engineered through the production of a dormant chemical defense system. The root inherently contains the specific precursors required to generate hydrogen cyanide, effectively storing a lethal respiratory toxin alongside its valuable starch reserves. These precursors are linamarin and lotostroline, stable molecules classified as cyanogenic glycosides. In the undisturbed living plant, these compounds act as a latent threat, sequestered in cellular vacuoles designed to remain separate from the enzyme linamaras. The lethal potential of the tuber creates a high-stakes biological scenario. The moment the physical integrity of the root is compromised, whether by the mandibles of an insect or the blade of a harvester, the cellular compartmentalization triggers a catastrophic failure. The rupture allows the linamarase enzyme to mix freely with the glycosides, initiating a rapid enzymatic hydrolysis. This reaction liberates hydrogen cyanide, a volatile gas that halts cellular respiration in mammals. The plant functions much like a two-part industrial epoxy. The components are inert while separated by a physical barrier, but once that internal wall is breached, the reaction is immediate and potent. The intensity of this defense varies significantly depending on the genetics of the crop. Sweet cassava varieties generally maintain lower baseline levels of these compounds, often requiring less aggressive intervention to become edible. However, the bitter varieties, which are frequently more robust in difficult farming conditions, contain concentrations of cyanogenic glycosides high enough to be fatal. Improper preparation of these bitter strains often leads to kanzo, a permanent paralytic disease prevalent in regions forced to rely on shortcut processing methods. The effective neutralization of this threat demands a processing chain that prioritizes chemical exhaust over culinary speed. To render the root safe, you must deliberately facilitate the release of the poison before ingestion. The most effective protocols begin with extensive mechanical disruption. Grating or crushing the raw root is not merely for texture, it is a requisite step to shatter the cell walls and force the enzyme into contact with the glycosides. This intentional mixing triggers the cyanide release while the pulp is exposed to the air, allowing the volatile gas to dissipate harmlessly into the atmosphere. Industrial production treats this detoxification with the same seriousness as hazardous material handling, utilizing automated graders to ensure the cyanide content drops below safe limits. Following this disruption, the pulp undergoes a regimen of soaking and fermentation. Submerging the material allows the soluble glycosides to leach out into the water, while the fermentation process lowers the pH, further destabilizing the toxic bond. Research indicates that the simple application of heat, the standard method for most vegetable preparation, is dangerously insufficient for bitter cassava. The physics of the process work against the cook in this scenario. If the root is thrown into boiling water without prior physical processing, the intense heat rapidly denatures the linolmerase enzyme before it can successfully break down the toxin. This effectively inhibits the release mechanism and locks the cyanogenic glycosides inside the cooked starch, creating a hidden reservoir of poison waiting to be released by the consumer's own digestion. Consequently, the volatile compounds must be allowed to escape through grating and fermentation before the product is ever introduced to heat. This elaborate processing chain transforms a potentially lethal root into a stable, caloric staple, proving that safety is achieved not through avoidance, but through the rigorous application of chemistry in the kitchen. Yet, while cassava demands a specific mechanical intervention, other botanical families present a different challenge, where the line between a common vegetable and a toxic hazard is blurred by a confusing system of taxonomy. The chaotic classification of the nightshade family often creates a botanical minefield for the uninitiated consumer. While the potato relies on solanine, its cousins, the tomato and the tomatillo, operate under a different, yet equally rigorous, alkaloid profile. The primary compound here is tomatine, a defensive chemical synthesized to deter herbivores from consuming the plant's infrastructure before the seeds are viable. This alkaloid is not evenly distributed. Rather, it is strategically concentrated in the vegetative parts of the organism, the vines, the leaves, and the stems. Ingesting these structural components, or indiscriminately juicing the vines alongside the fruit, introduces a significant load of toxins to the digestive system, capable of causing headaches, dizziness, and severe gastrointestinal distress. The maturation of the fruit represents a deliberate chemical disarmament. As a red tomato ripens, the plant actively degrades the tomatine content, interpreting the color change as a signal that the seeds are ready for dispersal. However, the green tomato operates as a fortified bunker. Because it is immature, the plant retains higher levels of alkaloids within the flesh to prevent premature consumption. This creates a distinct culinary risk profile. The popular preparation of fried green tomatoes is not merely a preference for tartness. It is a method that relies on the specific selection of varieties and the application of heat to mitigate the inherent bitterness and reduce the toxicity of the unripe tissue. Without this thermal processing, the raw green fruit remains a source of potential illness rather than nutrition. The tomatillo presents a more complex anatomical challenge, utilizing a physical barrier known as the calyx, or husk, to protect its development. This papery shield envelops the fruit, trapping a unique chemical defense mechanism against the skin. If you peel back the husk of a fresh tomatillo, you will encounter a sticky, viscous residue coating the surface of the green fruit. This layer is not a product of decomposition, but a concentration of defensive chemicals designed to repel insects and bacteria. The preparation of this vegetable requires a crucial hygienic step, the thorough washing of the fruit to dissolve and remove this sticky film. Cooking the tomatillo without first scrubbing away this residue incorporates the defensive irritants directly into the meal, altering both the flavor profile and the safety of the dish. It is also vital to distinguish between the photochemistry of the potato and the ripening process of other nightshades. A common misunderstanding suggests that any green nightshade is laden with solanine. However, plant biochemistry experts clarify that while the potato synthesizes solanine when exposed to light, other members of the family, specifically eggplants, peppers, and tomatoes, do not produce this specific neurotoxin even when they are green. An unripe bell pepper or a green eggplant does not pose the same neurological threat as a green potato. The danger in tomatoes and tomatillos is strictly bound to tomatine and the consumption of the non-edible vegetative parts, rather than a reaction to sunlight. The selection process, therefore, demands an eye for the visual stages of biological maturity. For the tomatillo, safety is signaled by the condition of the husk. A dry, brown, and peeling calyx indicates that the fruit has reached its chemical peak and is ready for harvest. For the tomato, the transition from green to red marks the degradation of its defensive arsenal. By respecting these distinct phases of development and strictly avoiding the consumption of the vines, even when the produce is visually marketed as on the vine, you ensure the meal remains safe. Yet, identifying the inherent chemical state of the vegetable is only half the battle. While the plant manages its own toxicity during growth, a new, invisible spectrum of hazards emerges the moment the produce leaves the safety of the field and enters the supply chain. The danger profile of vegetables shifts dramatically the moment the crop is severed from its roots. While the previous examples dealt with inherent genetic defenses programmed into the plant's DNA, the final category of risk stems from external environmental contamination. The plant itself may be biologically perfect, yet it serves as a carrier for a microscopic biological army introduced long after the seed germinated. Pathogens such as Listeria monocytogenes and E. coli AO157 H7 do not occur naturally within the cellular structure of healthy vegetables. Instead, they are introduced through the logistics of agriculture. Contaminated water used for irrigation or animal manure applied as fertilizer during the high-growth season. This turns the exterior of the produce into a potential vector for severe illness, regardless of the plant's internal safety. The complexity of this threat is amplified by the physical anatomy of specific crops. Rough-surfaced produce, such as cantaloupes and netted potatoes, present a topographical challenge that favors the pathogen. The microscopic crevices and textured weaves of these rinds provide ideal microhabitats for bacteria to adhere and form resilient biofilms. These microorganisms anchor themselves into the ridges of the skin, resisting simple rinses. A common operational error in the kitchen involves treating the rind as a disposable packaging layer that requires no sanitation. However, the physical act of slicing through an unwashed melon creates a direct contamination corridor. This phenomenon is known as internalization. As the knife blade penetrates the contaminated surface, it acts as a mechanical shuttle, dragging pathogens from the exterior rind and driving them deep into the previously sterile, nutrient-rich flesh. Even if the consumer never touches the skin, the very tool used to prepare the meal has successfully inoculated the food with bacteria like E. coli. Therefore, the protocol of washing produce must be applied universally, irrespective of whether the skin is intended for consumption. The surface must be sanitized to protect the interior. Once the produce enters the home, the storage environment introduces a secondary, paradoxical risk. The modern refrigerator is designed to suppress bacterial growth through low temperatures, a strategy that is effective for the vast majority of food spoilage organisms. However, Listeria monocytogenes is a biological anomaly that defies this thermal rule. It is uniquely adapted to thrive in cold, moist, and dark environments. While other bacteria enter a state of dormancy, Listeria continues to proliferate, turning the vegetable drawer into an active incubator. This capability poses a significant challenge, as the standard practice of cold storage does not guarantee the biochemical stasis of the pathogen. Data analysis reinforces the severity of this invisible threat. A review by Consumer Reports, covering recall data between 2017 and 2022, identified distinct patterns of vulnerability across the supply chain. Leafy greens, onions, papayas, peaches, and cantaloupe were frequently implicated in safety alerts. Most notably, prepackaged salad mixes and romaine lettuce were among the most commonly recalled items. The complex surface area of leafy greens makes them difficult to sanitize industrially, and the sealed environment of a plastic bag creates the humid conditions that favor pathogen survival. In this dataset, mixed greens contributed to the highest frequency of fatalities, highlighting the lethal potential of these ready-to-eat conveniences. Mitigation of these risks requires a shift from passive storage to active intervention. Relying solely on cold air is insufficient. For firm produce like apples, pears, and bell peppers, the only effective method is a rigorous mechanical scrubbing under running water. The friction of a clean brush or cloth is necessary to physically dislodge the bacterial load that water alone cannot displace. Finally, the process must conclude with thorough drying. Leftover moisture on the surface of a tomato or cucumber provides the hydration necessary for residual bacteria to multiply. By drying the produce with a clean towel, you remove the essential resource the pathogen requires to survive. Ultimately, whether dealing with invisible bacteria or visible plant anatomy, the boundary between sustenance and sickness is defined by how well you understand the biological reality of your ingredients. The distinction between a nutritious meal and a medical emergency relies entirely on the precise application of this botanical knowledge. It is crucial to recognize that the vast majority of produce is perfectly safe for consumption. By respecting the chemical defenses of plants, from the solanine in tubers to the cyanide in tropical roots, and maintaining rigorous processing standards, the actual risk of illness becomes statistically remote. You effectively neutralize inherent risks while retaining the nutritional benefits. The danger lies not in the nature of the plant, but in the absence of proper preparation. Assessing your current kitchen protocols against these biological realities ensures that every ingredient serves your health rather than compromising it. If you found this analysis valuable, don't forget to like, share, and subscribe to the channel for further insights.